Welcome back to the second part of DNA replication process. So, in this process, we are going to explain the main enzymes and proteins that are required for DNA replication. So, there is a question we have to answer, it, which is what are the main proteins and enzymes that are having essential roles in this process in DNA replication in cell division? So let's see. The first enzyme, which is more important in this process, is called topoisomerase. In eukaryotic, in human cell, it's called DNA topoisomerase 2, whereas in bacteria, it's called topoisomerase or also gyrase enzyme type 1, type 2, type 3. So this enzyme has three other kinds. The first part, which is called type 1 gyrase or topoisomerase, it breaks the DNA in only one strand of DNA. Whereas the type 2, which is present in the human cells, is responsible for cutting double strands. So this protein or this enzyme responsible for removing the tension, or it reduces the topology or coiling structure of DNA. But there's a question, how? So this type of enzyme has two different domains. The first domain is called nuclease, which acts as a nuclease enzyme, acts as a scissor, breaks the DNA. And the second domain acts as a ligase, so it's called ligase domain. So this activity helps the smooth progression of DNA replication and maintain genomic stability in human cell or bacterial cells. Now, for initiation of this process, there is a protein which is called pre-replication complex protein. And this protein, firstly, it moves, then, secondly, it recognizes a specific place in our DNA, in our genome, or in bacterial genome, which is called origin of replication. As in previous video, we explained in eukaryotic, there are multiple replication origins, whereas in bacterial cell, there's only a single origin of replication. So the pre-replication protein complex, with the help of other enzymes, they are acting as a helicase enzyme, opens the double-stranded DNA in order to start this process, in order to replicate the DNA itself. Now, the second one, which we include into the second stage, which is called elongation. There's a specific enzyme responsible for breaking down the hydrogen bonds between double-stranded DNA, and it's called helicase enzyme. So helicase enzyme breaks down the hydrogen bonds between guanine and cytosine, adenine and thymine, and it produces two single-stranded DNA. Now, these separated strands, they want to rejoining, because both they have free hydrogen bonds. So we have the second protein, which gives the stability, which gives the which protects for these two separated strands from rebinding, and it's called SSB protein, single strand binding protein. This single strand binding protein, firstly, it binds with the separated strands and it protects from rejoining, from rebinding. And the second important function for it, it protects the separated strands from nuclease enzyme. As we know, within the nucleus, there are multiple types of nuclease enzymes. They are breaking down the foreign DNA. So how our separated strands protect from these? By the help of SSB protein. Then SSB protein and helicase enzyme, both they produce two separated, they produce two separated single strands. Now, we need to start the synthesis of new strands for parent or old strands. The enzyme which is responsible for synthesizing and adding nucleotides for synthesizing new strands is called polymerase enzyme, DNA polymerase enzyme. But this enzyme cannot work alone. It needs what we can say small short nucleotides, sequence nucleotide. So we have another enzyme which is called primase enzyme. Some they said DNA primase or RNA primase. So it's primase enzyme responsible for producing or synthesizing a short single RNA sequence. Then after that, polymerase enzyme read and binds with this primer and synthesizing new strands by adding new nucleotides. So the DNA polymerase produces a replication fork, then the replication fork makes two new strands. The two strands are called leading and lagging strand. Leading strand is the continuous one, whereas the discontinuous strand, which is formed from 5 to 3 prime, is called Okazaki fragment, or we can say incontinuous or lagging strand. So now the last two important enzymes which are responsible for producing or making or synthesizing two new template strand is called or are called polymerase 1 and ligase. Let's see. The polymerase 1 firstly reads the new synthesized strands. That's the first one. And secondly, if there's any mistake, it acts as an exonuclease activity. It removes the errors. Then it fills the gap. And also it removes the primers. So it acts as a polymerase enzyme by adding nucleotides, fills the gap, synthesize or complete the new strands. And the last one, which is called which is called ligase enzyme. So the ligase enzyme links or sealing the Okazaki fragments or the incontinuous fragments. And finally, all together, they produce two new strands in order to replicate or in order to one cell becomes two cell. So finally, thank you for watching, and to receive more scientific videos, please subscribe to our channel. And if you have any questions, leave a comment down below. Until the next video, bye.